All right, you're all engineers. I need something that will come along to a particular DNA sequence and cut it at that DNA sequence. Pick some sequence of letters. I want you to be able to cut the DNA there. How would you go to the engineering department and design something to cut DNA at a particular location? Any takers? Scissors. Sorry?
Scissors.
Scissors, very small scissors, right? Because the scissors have to break a sugar phosphate backbone, so they're going to be very little. Yes? That would be nice. You need something that matches up with the bases. And somehow, if you could get something that could recognize the bases, what would be good at recognizing bases? Maybe there's an enzyme. So what you should do is design an enzyme. You go off to the engineering department and say, design me an enzyme, please, that will cut at this particular sequence. Okay, and so that's what you do. And the engineering department would say, I have no idea how I would make an enzyme that recognizes this particular sequence and cuts there. They would say, go ask an expert. So that's what you do. You go ask an expert. Who's the expert?
You.
I'm not the expert. The expert is bacteria. Bacteria have been at life for much longer than we have. Turns out, not just that they actually evolved earlier, but remember the generation time of bacteria, 20 minutes, if we're talking about the E. coli bacteria. The generation time of most of you, 20, 25 years, something like that. They've had a lot more generations. Anything you want to know, you sit at the feet of bacteria and you ask. Now, bacteria don't actually have feet, but that's another question, okay? So you sit at the feet of bacteria and you ask, and it turns out that bacteria have exactly what you want. Bacteria have a protein that can recognize a specific sequence and cut at the specific sequence. Pretty cool. The enzyme we're talking about is called a restriction enzyme, and we'll see why in a minute. If I have a sequence, let's see, 5 prime, A, G, C, T, A, G, A, T, T, C, T, T, A, C, C, 3 prime. Going back in the opposite direction, what are we going to have? We're going to have a G, G, T, A, G, A, T, T, C, T, A, G, C, T, 3 prime. That'll be the sequence. It turns out that E. coli has an enzyme that will recognize this sequence. GAATTC. What is it on the other strand? GAATTC. That's kind of cool. It's a palindrome. It's not exactly an accident that's a palindrome because the enzyme is a dimer and therefore it recognizes in both directions. Anyway, it recognizes this palindromic sequence. And what it will do is, actually, it's a reverse palindrome. It's in opposite strands. It's a palindrome. This is the recognition sequence. And what the enzyme will do is the enzyme will cut the sugar phosphate backbone here, and because it's symmetric, here. And what you will get are 2 fragments of DNA, A, G, C, T, A, G, T, C, G, A, T, C, T, T, A, and A, T, T. You'll get 2 pieces, 5 prime, 3 prime, and you'll have a hydroxyl group here. You have the little phosphate group here, 5 prime. You have your phosphate group here and your hydroxyl there. And it cuts it very nicely. This enzyme is in E. coli. It was found in E. coli strain R. It was the first enzyme found in E. coli strain R. First restriction enzyme found in E. coli strain R, and therefore this very friendly enzyme that cuts and does this is called E. coli R1. That's the name of this enzyme. E. coli is the bacteria, strain R, number 1.
What is it named?
The naming is pretty straightforward. We could make the naming more complicated if you'd like, but the names actually make sense. What doesn't make sense is why E. coli has this enzyme. One hypothesis. E. coli evolved this enzyme over the course of 3 billion years so that when molecular biologists got to the point that they needed to be able to cut and paste DNA, it would be there.
I like that one.
It's a nice hypothesis, but I don't think it's very convincing. Why? Well, horizontal transfer from what? Why?
Well, mammals, E. coli need a way to share their genome.
Yeah, but cutting up DNA. Why does E. coli have an enzyme that cuts DNA? What's the advantage?
Circular?
Sorry?
It has circular.
It has circular DNA.
Wants to unfold it.
Well, yeah, maybe it's good for the E. coli. It cuts open its DNA. If you ask me, having your DNA in a big circle and then having an enzyme that goes cutting it up seems dangerous. Yes?
Viruses or bad DNA that they want to take out.
Oh. What if E. coli is worried about getting infected by a virus and is actually a defense mechanism? What if it's kind of an immune system, so to speak? Pretty clever. Viruses have been attacking bacteria for a long time. Bacteria are no fools. They evolve ways to fight back, and a DNA cutting enzyme is an answer. So why have restriction enzymes? It's a defense mechanism. It's a defense mechanism that what it does is it restricts viruses from growing. That's where the name restriction enzyme comes from, is they restrict the ability of viruses to grow in that E. coli cell, because when the virus comes into that E. coli cell, here we go, here's our E. coli cell. This virus attaches itself to the surface here. It shoots in some viral DNA, and our enzyme comes along, our restriction enzyme, looks for this site, GAATTC. How often should you find a GAATTC site? Let's see. What's the probability of a G? 1 in 4 if the bases were all equally likely. So a G is a 1 in 4 times an A times an A times a T times a T times the C. It should be 4 times 4 times 4 times 4 times, it should be 1 in 4 to the 6th. 4 to the 6th is about 4,000. We'd expect 1 every 4,000 bases. Most viruses will have such a site. Bingo. It cuts up that virus's DNA, and it's pretty smug about it. Any problem with this idea? Yes.
What if they cut their own?
Uh-oh. So wait. So E. coli has a genome that's sort of more on the order of 4 million bases. And so if our enzyme comes along and cuts the E. coli DNA, it'll cut it at 1,000 locations. Maybe E. coli evolves so it doesn't have any of those GAATTC sites. By the ones that didn't have it, doesn't sound so plausible. Any other suggestions?
They have another mechanism so they don't cut their own DNA?
Bingo. They have another mechanism so they don't cut their own DNA. Safety scissors. No, it's not safety scissors. It turns out that when you have, let's go take a look at this here. when you have GAA-TTC, when you have this site, what happens is there is a second enzyme that comes along, and the second enzyme, this one was a restriction enzyme. This second enzyme, we'll put an enzyme here, and we'll put our other restriction enzyme here. This enzyme adds a methyl group. It sticks on an extra methyl group at that position. What it does is right over here, it'll add a methyl group. This enzyme, this green enzyme, is called a methylase. So there's a restriction enzyme here, and there's a methylase with a matching sequence. So E. coli first protects all of its own Echo R1 sites. We'll call this GAA-TTC an Echo R1 site. All of its own chromosomal Echo R1 sites are protected by the methylase. The enzyme, the restriction enzyme, won't cut, won't match, won't recognize if there's that methyl group there. Pretty clever. Restriction methylation pairs. And the critical thing is the bacteria has an enzyme, it cuts at the sequence. The bacteria has an enzyme that methylates at that sequence. It protects its own DNA by the methylation getting there. And when the virus comes in, the restriction enzyme just gets there first. That's it. Very simple. By the way, when the bacteria replicates its DNA, and it makes a new strand of DNA, will there be methyl groups on both sides? There'll only be methyl groups on one side. But it turns out that's enough. Methyl groups on one side are enough to protect, and therefore it helps the chromosome. As long as the chromosome was fully methylated, then when it starts replicating, it's now only half methylated on the old strand, but not the new strand. That's enough protection, and that gives the restriction enzyme won't cut, and the methylase has time to get there and tidy it up and put the other methyl group on. It's actually really clever. Bacteria are a lot smarter than we are. Okay, so now, this is not the only restriction enzyme. It turns out there are a lot of restriction enzymes. There are restriction enzymes, for example, of all varieties. Echo R1 cuts at GAA-TTC. This guy here, BAM-H1, cuts at GGA-TCC. Different strain of bacteria makes a different enzyme, cuts in a different way. This guy here, this is called BAM-H1. Bagel2 is what we call this guy. Bagel2 cuts at AGATCT. BCL1 cuts at this sequence. Well, look at that. TAA1, actually, instead of making a staggered cut like that, makes a cut like that. Echo 53K1 cuts right down the middle with no staggered anything, just smack down the middle, et cetera, et cetera. It's amazing. There are enzymes. Bacteria have come up with all sorts of enzymes that cut at all sorts of sequences in their wars against viruses. Virtually everything that can be done gets done. The bacteria have come up with solutions of all sorts. There are staggered cuts this way with a 5' end overhanging, staggered cuts that way with a 3' end overhanging, cuts in the middle. There are sequences, usually they're palindromes, they're not always palindromes. Sometimes a couple of the bases in the middle don't matter. Sometimes they cut at 4 base sequences, sometimes at 6 base sequences, some at 8 base sequences. There are some that cut at 5 base sequences. They've had a long time to work this out, yes. Now, how do you get a restriction enzyme? Well, of course, it's biochemistry. You're going to take the E. coli strain that has this restriction enzyme, you're going to grow up a lot of that strain, you're going to add the cells to liquid, they'll grow and divide, you'll grind them up and you'll purify this enzyme. And you have an assay, it'll cut this sequence. So you hand it the DNA, you purify out proteins, you see which fraction is able to cut it, and then that's how people got Echo R1. And in old times, ancient days, when ancient scientists in the 1980s 80s, for example, wanted EcoR1, they would have to grow up a bunch of E. coli and purify EcoR1. And if they wanted BAMH1, they'd have to grow up the appropriate bacteria and they would have to purify it. Nowadays, that doesn't have to happen. If you want EcoR1, where do you go? You go to the catalog. So there's a catalog. And in the catalog, they have restriction enzymes. All sorts of restriction enzymes. Let's see what we got here. Oh my, let's see. EcoR1, right there. EcoR1. Let's see, what is it going for? You know, it's 10,000 units for $53. That's pretty good. We got you know, BAMs, and we got, oh, look at this. It's just pages and pages and pages of restriction enzymes. And if you have a credit card and you order before midnight tonight, you can get EcoR1 or BAMH1, or there's probably a discount on PST1 or whatever. And that's what molecular biologists do now, is if they need a particular restriction enzyme, it's in the catalog. Okay. It's, of course, really online. You don't even need the catalog. You can just go online and order a restriction enzyme. Okay, now, that was cut. What about paste? Yes, please.
Different species of bacteria have a different...
No, most species have one or maybe two. They don't have thousands, and they all just have different restriction enzymes. I mean, it's a garden of wonderful different tools for the molecular biologist. And some companies like New England Biolabs go out and purify new strains and identify new restriction enzymes all the time. Yes?
Why do we need so many in the catalog?
Well, what if I want to cut at this sequence or that sequence or this sequence?
Okay, so
Depending on where I want to make a cut, I have a lot of choices. Absolutely. But now, having cut DNA up, I want to paste DNA. So now I've got to take pieces of DNA and I want to paste them together. So remember, we had I'm just going to simplify our sequence now. So it looked like this, and we had a GAATTC. So that means on this strand it'll be a TTAA, and we're going to have a 5' phosphate here, and we'll have our little hydroxyl there. And we've got another piece of DNA that matches it, AATT. And it's got a little phosphate there and a little hydroxyl there. And it might be that these were matching pieces that came from here. But it could equally well be that this is a piece of E. coli DNA, and that's a piece of zebra DNA. Why not? So we have to go to the catalog. Sorry, no, we have to go to the, we have to go to the engineering department and say, can you please make me an enzyme that can paste together DNA, that can catalyze the reaction of rejoining the sugar phosphate backbone. What'll happen is, if I add these pieces of E. coli DNA and zebra DNA, they'll hybridize to each other by Crick-Watson base pairing. They'll land on top of each other by Crick-Watson base pairing, but that's hydrogen bonding. They aren't covalently bonded to each other yet. We need to come along and we want to connect up covalent bonds along the sugar phosphate backbone. And we're going to want us an enzyme to do that. So we go to the engineering department, we say, please make an enzyme that'll take these pieces of DNA. And the engineering department says, I don't know how to do that. And they say, go ask an expert. And who do we ask? Bacteria. Question?
DNA ligase do this?
yeah.
Well, as it happens, we have heard about an enzyme that does this. And that enzyme is DNA ligase. Why do we have DNA ligase? We got to rejoin these lagging strands of DNA during replication. Bingo. We actually have an enzyme. Very good. DNA ligase, which we encountered when we talked about DNA replication, now turns out not to be just an interesting fact about DNA replication, it turns out to be a tool for molecular biology. Because we can use DNA ligase ourselves. And of course, how do you get DNA ligase? You grow up a lot of bacteria, you grind it up, and you purify DNA ligase. At least that's what ancient scientists did in the 1980s. But today, where do you get DNA ligase? Catalog. It's in the catalog. So you get it out of the catalog and you have DNA ligase, and you can ligate together anything with anything. Yes?
Are there no problems with, joining zebra DNA and E. coli DNA?
Are we talking about technical problems in joining zebra DNA and E. coli DNA? Ethical problems in joining zebra DNA? Elaborate on what kind of problems we have here. As a matter of chemistry, of rejoining a sugar phosphate chain, it's not problematic at all. If you handed ligase from E. coli, it will join fragments from E. coli, it'll join fragments from zebras, it'll join fragments from giraffes to zebras. It's an enzyme. These are pieces of DNA. It doesn't care where the fragments came from.
But you don't end up with like a weird mutant of some kind?
Well, I don't have anything. I don't have a mutant yet. I just have pieces of DNA in a test tube. Remember, when I take pieces of DNA and I've cut them, and then I paste them back together in some order, they're just pieces of DNA in a test tube. They're not yet a mutant. A mutant is an organism. This is just a piece of recombinant DNA. Recombinant DNA because I've recombined it in some new fashion. So this would be, when it's joined up, a piece of recombinant DNA. Now, you can talk about what we might do with that piece of recombinant DNA, put it in an organism, but at the moment it's just a piece of, it's just a DNA strand. And the truth is you can't tell the difference from any other kind of DNA strand unless you happen to know that this sequence was from the zebra and this sequence was from E. coli. All right, stop for a second and test your understanding about the cutting and pasting of DNA.